I think you have to do the thing first.
What's that?
Get rid of the.
What do you say? Yeah. I don't think Eric was here on just
From
because it's sitting up here. So it's my great honor to introduce Caleb here. Yes, Caleb Dvorak. He previously actually lived here in St. Louis. He lived in Tower Grove South. He worked at the Botanical Gardens, and he did his education in Hawaii on this particular subject of orchid flasking. Caleb is the person that we go to for our particular fasting needs, and he's here today to talk about all the steps of that process and give you guys a little bit of information about it. So thank you very much.
All right, how y'all doing?
I don't do anything.
So as you said, my name is Caleb Dvorak. I'm a bit of a laboratory guy. I'm used to spending all day in a windowless room by myself, so please bear with me. Public speaking is right up there with hungry alligators and tornadoes. So let's get started.
This is what I chose for a living, for an education. I got my bachelor's degree in horticulture from Iowa State University, and then I did a student exchange in Hawaii where I was introduced into tissue culture. I fell in love with it, so as soon as I completed my bachelor's degree, I went down there and got a plant tissue culture certificate. While I was down there, I earned food money by working at a local laboratory where we grew all kinds of amazing tropical plants in the laboratory. And that's pretty much when I first heard of orchids. And then as soon as I got my certificate, I came back to the mainland, as they say, and worked in a very large laboratory just outside of Chicago for Ball Horticultural Company. And we were pumping out about 500,000 ornamental tissue culture plants.
Low. So that was a good trial by fire.
After that, we moved to St. Louis, and I worked at the Botanical Garden. I helped them set up a laboratory, so we have a tissue culture laboratory at the Botanical Garden now. And while there, I got to grow some amazing native orchids that grow around here, and that's kind of what I still enjoy growing once in a while. Not too many epiphytes, but I do enjoy the natives, the ones that grow on the ground. And then finally, my family moved to Texas, and I built my own laboratory down there, and now I run a little business creatively called Flask Factory. So that's what I do now. So we're going to talk about flasking orchids from seed. You got a seed pod, you got the mother flask, that's right after germination. You've got an intermediate flask here, and that's a flask that's just about ready for shipping out to one of my customers. So some of the topics we're going to cover today is what is flasking, the science behind orchid seeds and why they're well suited for flasking, the laboratory equipment and materials that you may need to do flasking, flasking media and how you prepare it, some of the sterile technique you'll need to use to keep everything clean, sowing orchid seeds, deflasking orchid seeds, and then if you're wanting to send some seeds to someone to flask them for you, hopefully me, how to harvest and ship them in the best way possible. Is anybody here familiar with flasking? Ever heard of it? A few of you? So just about all of the orchids that you have growing probably went through a flask at some point. It's the process of growing orchids from tiny dust-like seeds in a sterile environment using a nutrient-rich agar media. Agar is kind of like gelatin, and that encourages germination and early growth of the plants. This process typically takes place in a laboratory, but it can be done at home with the right skills and equipment. I'm going to try to briefly go over some of that, so if you want to try it yourself, hopefully this will help you. Flasking is another term for the tissue culture or micropropagation of orchids. And tissue culture, micropropagation, that's kind of my background, so sometimes I may slip into using those terms, but just know that that's flasking. So orchid seeds are different than most seeds. Normally, plant seeds, like this kernel of corn here, contain an embryo along with a whole bunch of endosperm. All that white stuff is endosperm. that provides food for the embryo to germinate so that it can grow into a seedling that's capable of photosynthesis. Photosynthesis is how plants obtain their energy, but seeds can't really photosynthesize, so they use that endosperm to grow into a little plant, and then they can have their own energy. Orchid seeds, on the other hand, are tiny and dust-like. They contained just an underdeveloped embryo surrounded by a thin seed coat that's only about 1 cell thick. They don't contain any endosperm, so they don't have that energy to give them a boost to be able to grow out some leaves and photosynthesize. That right there is a 1 millimeter long orchid seed, about this big. That's pretty big for an orchid seed, frankly. Usually they're much smaller than that. So this smallness of the seeds means excellent wind dispersal, but the seeds must land on a suitable surface that harbors an appropriate symbiotic fungus in order to germinate. That's a pretty hard thing to do, so orchids produce thousands and thousands of seeds per pod in order to increase the chances of success. So there's some orchid seeds next to a little scalpel to give you an idea of how big they are. There they are under a microscope. If you've ever had a really nice vanilla ice cream and there's these little flecks inside the vanilla ice cream, those are actually orchid seeds from the vanilla orchid. So in nature, a seed has to, like I said, land on a suitable surface that harbors the compatible symbiotic fungus. The fungus actually infects the seed, and then the seed flips the script and it captures the fungus, and then it eats it. It digests it for energy. And this sort of replaces the role of the endosperm, and it provides the seed enough energy to germinate and grow into a small structure called a protocorm. And as the orchid continues to grow, the fungus will often hang around and continue to be present in the plant's root system as something called a mycorrhizal fungus. Mycorrhizal just means like the fungal zone of a plant. So here's sort of the stages. You can classify it into different stages of growth, but the little red dots are the fungus, and the cells of the orchid capture it into a little structure called a peloton. It almost looks like a ball of yarn, and the yarn is the fungus, the hyphae, that it has captured and is digesting and using for energy. So once an orchid grows leaves, it begins to rely more heavily upon photosynthesis, much like a normal plant, and the fungal relationship becomes a little less important as a source of carbon. It uses photosynthesis to obtain carbon and build its body. But of course, there are always exceptions. Some of those are actually right here in Missouri. If you go out into the woods, have any of you ever seen a coral root orchid? They're kind of neat. Corallarhiza, coral root orchids, and I believe Hexalectris may also be found here. They rely entirely on fungi as a source of carbon. So they don't even have leaves or the ability to photosynthesize. They rely entirely on fungi to obtain their energy. They're kind of neat.
Why did they give the fungus?
I'm sorry?
Why did they give the fungus?
That's kind of debated right now. They call it a symbiotic relationship, but it may be more parasitic, where the fungus is actually parasitizing, or the orchid is parasitizing on the fungus. I believe they're trying to track carbon isotopes from the fungus and the orchid and see which direction those isotopes are, to see which direction the carbon is flowing. And that can kind of tell you who's getting what from who. And I'm not sure, but I think kind of right now they're thinking that the orchid is kind of getting the better end of the deal. Hey, they've figured out how to get us to take care of them, so. So you can do symbiotic culture. You can use the fungus to grow orchids. It's not the normal way, but I did a lot of that at the botanical garden. First, you'll collect some roots of orchids, then you macerate the roots, you grind them up, and then you use the root juice and you spread it onto an agar that has what you call a fungal isolation media. It's a really low nutrient media, and it includes things like antibiotics, because orchid roots are disgusting and they grow all kinds of things that you don't want. So you're trying to eliminate bacteria. Once that grows out, you inspect it under a microscope, and you're looking for just the right type of fungus that's branching in just the right way. It's very tedious work, and then you try to isolate it under the microscope with the tip of your scalpel, and then pluck it out and put it on a new, richer agar media and grow out that fungus. And then once it's bigger, you can sort of confirm if you've gotten the right thing, the right fungus. And then you test it with the seeds, preferably seeds from the plant that you just isolated the fungus from. And then you can, if it works and they germinate, you can grow them on. Yes?
How do you keep from just growing an amazing crop of mold on your
So after you spread the root juice onto the media, you usually have a few days before things get out of control. And so you're looking under the microscope and you're seeing just that early growth from the fungi and bacteria, but you ignore those. And like I said, you're sort of looking for the right sort of hyphal branching patterns. It almost looks like a snowflake rather than a bunch of straight lines. They also tend to be a little less vigorous than something that's like a mold. A mold is going to take over, so you kind of have to be quick. At the same time, it's tedious. It's tricky. You just kind of have to get a feel for it. Yes?
Is there a certain type of fungus?
Yeah, so
Or is it really any?
no. So the common ones up here are called Ceratobasidium and Tulisnella. I believe I may have been, don't quote me on this, but I may have been the second person to find a, oh my gosh, now I'm going to blank on what it's called. A third type of fungus that's a little less common, especially up here in temperate regions. But I isolated that from a calopogon, and it works really well to germinate calopogon. Sebacina, that's the third one.
Follow-up question to that. Do different types of fungus react in different orchids and like produce orchids to grow in a certain way from fungus to fungus?
Not necessarily. Like some you'll see higher germination rates but slower growth. Some you'll see like rapid growth, but it murders most of the seeds, and the ones that are strong enough grow really well. They're able to overcome the fungus, right? But as the plant grows, I don't believe you'll see any like difference in the morphology of the plant or anything.
Yeah, like one fungus wouldn't turn in a different color
No,
or like germination.
no. So there's pros and cons to doing symbiotic culture. I, of course, always advise anyone against it, especially if you're looking at native orchids, because you are doing an invasive procedure on the orchid. You're ripping roots off of an orchid, and a lot of our orchids are rare. They only grow in a really pristine environment. So try to avoid it. Best left to, I hate to call myself a professional, but best left to professionals. You have to get faster germination, maybe lower germination percentages, but they grow faster. The growing medium for them is really simple. It's just water, ground up oatmeal, and agar. The plants may be better adapted to like stressors in the environment when they have their fungal partners. There are conservation advantages, which is sort of what we were focused on here at the botanical garden. You get to reintroduce the fungus to the environment if you're trying to grow native orchids. So let's say we find some rare native orchids and we want to establish another population in a suitable area. You can collect seeds from these rare orchids. You can grow them asymbiotically without the fungus, introduce them to the new area, and maybe those plants do really well. They bloom, they produce seed, they release the seed, but there's no fungus to germinate the seed, right? But if you have that fungus from the start, when you grew your plants, when you plant those plants into that new environment, the fungus grows out into the new environment. And so then when they produce seeds, those seeds land on the suitable environment, harboring the appropriate fungus, and you have a much better chance of establishing a long-term population. It also just helps you with research, like identifying which fungi go with which orchids and having all that information is good for future conservation efforts. There are cons. Fungal isolation is a difficult process. It's tedious. It takes some skill. I spent a couple days at a laboratory learning it from someone who's done it for decades. And then I had to try it a lot before I got it before I learned how to do it well. You must sacrifice the roots of an existing and possibly wild or rare orchid plant. So it's invasive. Orchids are very often selective about which fungal strain they use for germination. So you may isolate some really good fungi from the roots of the orchid, but they might just use those in the adult stage, but they don't use them for germination. So orchids will sometimes kind of switch which fungus they use at which life cycle. So you might isolate something good, but it doesn't germinate. Or you might isolate something that germinates great, but it doesn't really help the orchid grow at a later stage. There are also legal and environmental concerns. These fungi are very closely related to many crop diseases. So there are a lot of regulations about moving them. Sometimes you can't even move them from state to state. You definitely can't move them from country to country unless you have a certified quarantine-style laboratory with all the right equipment and certifications and everything. Yes?
When you say you must sacrifice the roots of an existing plant, is that all the roots, or is there enough to have the plant continue to grow and you're just taking maybe half of them?
Right. If you're careful, you can slowly excavate next to the plant. You might be able to pluck a root or two. Often the root that contains the fungus, the orchid roots are nice and pale white, at least for a terrestrial orchid. And then you may see some like brown discoloration on it. That's a good indication that that one contains the fungus. So you might select that root and leave another one. If it's a nice thick, starchy root, right, like a carrot, usually there's not a lot of fungus in it, and the orchid is really going to use that for energy. So you try to avoid those. You might take like a little lateral skinny root rather than the thick root. But if it's a big population of a very common orchid, it may be a little quicker to just take one or two from the population if there's enough and it's not a really sensitive species. Yes?
So if you are, say, you have a seed pod from a plant from Malaysia,
Mm-hmm.
do you have to use the roots from either the exact same plant, or can you use the roots from a plant from Peru? I mean,
Right.
do you have to use Cattleya roots to germinate a Cattleya plant?
It's an excellent question. Actually, I teamed up with a professor. We wrote a paper kind of on that. I should know the title of my own paper. I believe it's with Dr. Zettler. He's really good at this kind of thing. I learned a lot from him, so I have to give him a lot of credit. And he isolated a fungus. It's a Tulisanella calospora, and it's given the moniker 266, and they call it a super fungus because it's been shown to grow all kinds of different orchids. So some orchids are very specific. One of our native orchids will only grow with one specific kind of fungus. Other orchids are less picky. Some fungi are only really good for one or two orchids. Some fungi are good for a lot of orchids. It's really a kind of a trial and error. But you're trying to avoid introducing new fungi to brand new environments a long ways away. You don't really know how the fungus is going to interact with that environment, so you do try to do it with restrictions. We're not really going to go over in depth how to do this. I just kind of wanted to show it for showing the relationship between orchids and fungi, and how this is one way to do it, and then the more common commercially viable way is what we'll go into further. This is a little more, I guess, interesting, a little more novel. Not a lot of people understand how it works, and we do some of this stuff right here at the Botanical Garden, so I thought it'd be fun to go over. Speaking of, so this is asymbiotic seed germination. You're not using a fungus anymore. This is much more commercially viable, it's more common. So if an orchid can replace the need for endosperm in the seed, if they can replace that with fungi, then someone in the lab can actually replace the fungi with a nutrient-rich agar media in order to germinate seeds and grow on seedlings. So with small adjustments to the media, you can germinate seeds and then grow on thousands of vigorous orchid seedlings, and you can produce thousands of them from a single pod if you have good seeds. So the first media formulation for germinating orchid seeds was actually created by Louis Knudsen way back in 1922. Typical orchid media consists of carbohydrates, typically sugar, macronutrients that you're probably familiar with, nitrogen, phosphorus, potassium, micronutrients such as iron, copper, zinc, vitamins, amino acids, and then a gelling agent, typically agar. There's a list of the chemicals that, well, everything's chemicals, right? But a list of all the nutrients that are typically contained in orchid germination or just general orchid media. And yeah, we all feel totally capable of making that, right? I don't bother with that. Hardly anybody does. So maybe Dr. Madsen makes his own custom media. Yeah, that's awesome. But there are tissue culture supply companies such as Phytotech Labs based in Kansas. And they make creating media much easier. They make pre-mixed media powders. It contains basically that whole list. It's just a powder. You measure it out. You add water, a few other things. Really simplifies the process. And you know, people have been working on this since 1922, and a lot of times they can do a better job than most people. Maybe not Dr. Madsen. Okay. So this might be familiar from biology class. Photosynthesis. Plants use carbon dioxide, water, light. They give off oxygen, and they create glucose. Glucose they turn into more complex carbohydrates. They build their bodies with it, right? Respiration, that's what we do. We bring in oxygen. We eat food like sugar, and then we create chemical energy, heat, and carbon dioxide. Plants do respire, but photosynthesis makes up the larger balance. Plants, animals, right? But when you're growing them in a flask, it's a little different. Photosynthesis is autotrophy, that's making your own food. In a flask, they're being heterotrophs like us. They're eating food. They're also kind of doing some of both, and you'd call that mixotrophy. So in a sealed flask, orchids have limited ability to photosynthesize because they have limited access to carbon dioxide. It's a sealed flask. It's airtight. Instead, they become heavily heterotrophic, like us. They eat the sugar that's included in the media. Once they're eating, they're respiring, they're exhaling CO2. Once that CO2 is built up to usable concentration, then they'll use that with photosynthesis. They'll use it for photosynthesis, and they'll kind of switch back and forth depending on the gases that are contained in the flask. So if they're eating sugar that you provide in the media, why do we still use lights? We use lights when we're growing flasks. Well, they can photosynthesize if you include light. And while the photosynthesis is limited, it does increase their growth rates. The light helps them develop chlorophyll. And chlorophyll needs light to develop, and they're going to really need that once you take them out of the flask. They need to be able to photosynthesize once you take them out. Morphology, so the shape of the plants is not going to be right. The shape of their leaves, they're going to be stretched out. They're not going to look right if you're not providing light. It affects their physiology. Light ensures that they can synthesize the right hormones. Plants use hormones to grow in the right ways. They use light to develop the right metabolic pathways, etc. It's a bit over my head. And then for deflasking, light in the lab reduces the amount of shock that your orchids are going to experience once you take them out. It helps prevent sunburn, basically. They get a little tan. There's some Paphiopedilums that I took out of the flask. I was experimenting with some photography there. Yeah, it would look even better with the lights out, but. I appreciate that. Thank you.
This one.
YouTube. You can learn anything on YouTube, including how to take nice up-close photos. Theirs was for beautiful orchid flowers, but I thought, I've got flasks. Any questions on like the science behind it before we move on to the laboratory?
Has
Yes?
anybody ever tried to inject CO2 to increase growth in a flask at the same time? It'd be hard to do it sterilely, but.
They've done it with tissue culture. Can't remember. And I have the book like downloaded on my computer. But yeah, so they kind of use a sterile environment. They use big sealed like glass containers, and then they pump those with CO2. And then you need a certain gas exchange rate. And then they don't really include sugar in the media, so the plants are actually photosynthesizing, but in like a sterile environment. It's a really cool book. I'll try to look it up and I'll share it with you.
It's really popular in the aquarium hobby.
Yeah? Yeah, injecting CO2. Also, like with cannabis growing, they'll just flood the room with CO2, and it's plant food, right? So with the laboratory, we'll talk about lab layout, equipment, materials, media preparation, sterile technique, sowing, and transferring orchids in the flask. So if you're a nice big company, you can build, wow, office. That's weird. You can build a laboratory, and this is going to be your typical layout. You'll have your office for your computer work. Media prep is where you make your media. It's much like a kitchen. Then you'll have a transfer room. It's a really clean room with nice clean air where you can work on your cultures. That's throwing me off. Where you can work on your cultures without getting them contaminated. And then you'll have your culture room, which are shelves of light filled with flasks, windowless room. It'll, of course, be temperature controlled, controlled lighting, everything. This is a quick list of some of the equipment and materials you'll want for flasking. We'll kind of go through some of these individually, but you can reference this list if you're looking to do some of this on your own at home. One of your major pieces of equipment is going to be a laminar flow hood or a still air box. This is where you have a clean workspace with clean air. The air needs to be free of any kind of bacteria, viruses, any other microbes, the kind that we're breathing in right now. Gross, right? This allows you to work on your plant material, open up your sterile flasks, and they won't get contaminated. Just one little bacteria lands on that nice, delicious agar media, ruins it. One little spore from a fungus, it's ruined. So it needs to be super clean. This is a laminar flow hood that I built. This is a still air box. Still air box, pretty simple to make. It's like just a container that you can work inside. The idea is that the air is not moving. You spray it down with alcohol, and anything that's remaining in there will eventually settle to the bottom, and then you can work on your material without getting contamination. Excuse me. If you're working with flasks frequently, a laminar flow hood is where it's at. It's a lot more convenient, effective, and ergonomic than a still air box, but they are very expensive. If you're handy, you can build one yourself at a fraction of the price versus purchasing one. Instructions for building laminar flow hoods are pretty common on mushroom growing websites. Much of the process used for growing orchids is similar to the processes they may use for growing fungi. And those guys are very handy, and they love to share. So You can find instructions online for building your own. Don't confuse a laminar flow hood with a biosafety cabinet or a fume hood. If you're ever looking to purchase one, they work entirely differently. A laminar flow hood or a laminar flow bench pulls in air with a fan, and then it pushes it through a HEPA filter. It's basically a super heavy-duty filter that filters out any kind of contaminant down to a tiny, tiny size. It can filter out spores, bacteria, etc. And then it provides a nice, clean flow of air where you can open up your cultures and not worry about contamination. Next major piece of equipment is an autoclave or a pressure canner. So pressure canner helps you sterilize your flasks so that your media is 100% clean. An autoclave is basically a fancy pressure canner. They cost thousands, possibly tens of thousands of dollars. Just use a pressure canner. You're going to need sterile instruments, forceps, scalpels, etc. But those need to be 100% sterile. So the ways you could sterilize those is with a bead sterilizer. You could use an incinerator, or you can go old school. You could have an alcohol lamp. It just provides a continuous flame, and then you hold your instruments over there until they're red hot. Like a culinary torch for creme brulee works really well too. Just don't light it up right after you sprayed down your work surface with alcohol. Here's instruments. I like to have some long forceps, some fine-tipped forceps for little tiny work, and then scalpels are good for cutting open pods. I like to mount a magnetic knife holder on the side of my flow hood. It keeps them tucked out of the way, and then they're not really touching anything, so they stay clean. Volumetric glassware. Polypropylene glassware is what I use. It's cheaper. You can still run it through the autoclave or the pressure canner if you need to. And if you drop it, it won't shatter. Nice little digital scale. 0.01 grams precision is helpful. I apologize. The formatting on this, I must have opened the wrong file, but just bear with me.
It might be to switch from Google to PowerPoint.
Oh, okay. Next time. You'll want a pH meter and pH things to chemicals to adjust your pH. Just a simple handheld meter you can find on Amazon, works just fine. They're affordable. For adjusting pH of your media up, you can use potassium hydroxide, ammonia, baking soda works in a pinch. For adjusting pH down, vinegar, citric acid, hydrochloric acid. You don't have to go hardcore with the chemicals. Some of these you might just have at home. Then you want something to disinfect the surfaces of the plant material you're trying to put into culture, such as your seed pod. Bleach is most commonly used. I like to use something that is simply called sodium dichloroisocyanurates, NADCC. It's basically pool cleaner. It works much like bleach. It releases chlorine. It kills everything, but it's more pH neutral. It's gentler on the plants. And then you'll want a surfactant that helps your bleaching solution get right up against the plant tissues. You can use Tween 20 polysorbate. That's from Amazon. Or in a pinch, you can just use like dish soap. Then you'll need culture vessels, flasks. They should be appropriately sized to the stage of growth and should be as transparent as possible. You want to use materials that can be run through your pressure canner, such as polypropylene. That's PP number 5. If you do recycling, you've probably seen number 5. It's pretty common. That can usually withstand your autoclave, your pressure canner, or glass works nicely as well. You're going to need grow lights. We could spend the rest of the night talking about light. What kind of light? I'm sure you're familiar with light. You grow orchids. hopefully with sunlight, then you don't have to think about this too much. I like to use LEDs in the lab. They're most efficient and they produce less heat. When you have a laboratory with shelves full of lights up to the ceiling, the heat they give off can be substantial. And so to reduce the amount of heat, LEDs are best. You're going to want to get in that bright white range, 4,000 to 6,000 Kelvin. That's usually listed right there on the box when you go to pick them up at Home Depot or online. 4,000 to 6,000 Kelvin is pretty good. It's a little less blue than daylight, a little bluer than like a sunset. PPFD is simply what kind of light can the plants use. You want that in the range of 30 to 120. There are free apps that you could download on your phone. You can hold your phone under the light, see if you're in that 30 to 120 range. You can also use Lux, again, apps, 1,000 to 4,000. Lux does not tell you whether the light is actually usable by plants. You can have green light, the plants don't care about it, you can still have a high Lux value. That's why you're looking for something in that bright white 4,000 to 6,000 Kelvin range. Match that up with the right Lux, you're good to go. Try to evenly distribute your light. You don't want all the light hitting a few flasks and then the rest are in the shade. You kind of evenly distribute them. I have 4-foot shop lights placed about 2 feet apart. Works perfectly. Tissue culture media. Where are we at? All starts with good water, distilled water available at the grocery store. If you want to make a whole bunch of water, $50 reverse osmosis system that's portable, works great. That one's used for aquariums. I like to keep it simple. I get my premixed powder, and then I get some agar to solidify the gel. 668 from Phytotech is great at growing just about any kind of orchid. And then if you cut it in half, it's good for germination. You'll need to add back a little bit of sugar. But the real key are the complex organic additives. You might have coconut water, banana, potato, tomato. Orchids are weird. They like to eat these things, especially when they're in a flask. Really only when they're in a flask. Don't pour this on your plants. The type of additive and the concentration you use it at really depends on what kind of orchid you're growing, what stage it's at. Is it germinating? Is it growing on? It really just takes a lot of experimentation, A/B trials. Just keep trying until you find just what's right. Keep really good notes. You'll eventually figure it out if you do it enough. Let's cook. These are the basic steps for preparing media. You want to figure out how much media do I need to make. Then you'll create a nice little checklist. How much water, how much powdered media, how much agar, how much sugar, how much of your complex organic additive. You'll add half your water to the beaker, then you'll add your media powder, you'll add your agar, you'll add your sugar if you need it. You'll add your organic materials, and then you'll blend them up if you need to blend them. An immersion blender works great for this. Yes?
When you say add sugar, are you talking about adding the banana, or are you talking about table sugar?
So like that P668 powder comes with sugar in it. But if you're cutting it in half for germination, you still want the full concentration of sugar. So you're going to look at that list of ingredients. It's probably going to say 20 grams per liter of sugar. Once you make your germination media, now you've got 10 grams because you just cut it in half. So you're going to add back 10 grams per liter of sugar. It can just be table sugar, works fine. Does that answer your question? Okay. Once you've added all your ingredients, then you top off the water to the volume that you need. You mix it up. You use your nice cheap little pH meter. You measure it and you adjust it until it's between 5.6 and 5.8. Different orchids in nature love different pHs. Some grow in a bog, some grow on calcareous rocks. But in order for all those nutrients in your media to be available to the plant, it's almost always best to keep it right in that range, 5.6 to 5.8, no matter what kind of orchid you're growing. There may be exceptions. I have not run into them. You bring the media to a boil either on your stove or in your microwave. Don't put polypropylene beakers on the stove. This helps melt and activate the agar. The agar is the solidifying agent. It needs to come to a boil in order to be able to solidify. Then you pour it into your vessels. You loosely place the lids on before loading them into your pressure canner. If you tighten the lids, they could explode or they could implode. You probably won't know that it's happened until you open up your pressure canner and then you just see a big gooey mess. Not fun. If you're using thin-walled vessels such as like deli containers, then you're actually going to not pour the media into them before cooking. You want to stack them up, wrap them up in aluminum foil or a polypropylene bag, and cook them separately from your media. This is actually really space efficient as well. You can cook a lot more flasks in your pressure canner. And now I'm giving away the tools of the trade. Again. Then you're going to cook it at 15 pounds PSI. That's redundant. For 20 to 30 minutes. First, you need to add the appropriate amount of water to your pressure canner. You place the lid on. You remove your weight. You turn on the stove. You let it steam for 10 minutes. You want that steam coming out nice and steady for 10 minutes. You vent it. Then you put your weight back on, bring it up to 15 pounds of pressure. Cook it for 20 to 30 minutes. When 20 to 30 minutes is up, you turn off the heat, you allow the canner to depressurize, and then you open it and remove your vessels. You allow the media to completely cool and solidify before you use them. If you have the time, allow them to sit for a few days. This will help ensure that your sterilization process has worked. Otherwise, you're going to put your plant material in, and all of a sudden you have a bunch of bacteria. If you had just waited a few days, you'd see that you didn't run your pressure canner long enough. Any issues are going to show up in about a week. So if you can afford the time, just let them sit there for about a week in a cool, dry place before you introduce seeds or plants. This is just an example of me pouring out some Petri dishes for germination. It's the same general technique if you're using those deli cups with thin walls, cooking them separately. These Petri dishes came pre-sterilized, so I didn't have to worry about running them through the pressure canner. You wouldn't want to anyways. They're the wrong kind of plastic. They would end up a big sticky mess. Working in the flow hood here. Sterile technique. These are the skills that you'll need to be successful. Sterile technique is essentially just the practices that you use to prevent contamination of your cultures by microorganisms. Unless things are sterilized, you must assume that all surfaces, including your plant material and the air itself, are just covered in filth, bacteria, mold, spores, viruses, etc. Sterile technique allows you to do your work without contaminating your flasks. Flasks are very easily contaminated. They're nutrient dense. They're really good at growing orchids. They're even better at growing microbes. Microbes can compete with your orchid plants for nutrients. They can release toxins that will kill them. And they can also cause plant disease if you're oh so lucky to get just that right kind of bacteria. The best way to really think about sterile technique is separating things clean, dirty. Clean things. Sterilized media. It's been through the pressure canner. Everything's dead. Inside surfaces of your flasks, the tips of your instruments after you've sterilized them, the plant material that's been decontaminated with your bleach or your NADCC, the air that's inside your flow hood or inside your still air box, hopefully. Things that are dirty, the air outside your flow hood or your still air box, the surfaces inside your flow hood or your still air box. The surface of your flow hood, even though there's sterile air coming across it, just treat that surface like it's dirty. Even if you've just wiped it with 70% ethanol or alcohol, that alcohol kills a lot of stuff, but it doesn't kill everything. The exterior surfaces of your flasks, those are dirty. Your hands, your gloves, especially your breath, it's dirty. Your instruments or your plant material, if they ever come into contact with anything dirty, those are dirty. Clean, dirty, never the twain shall meet. It's all good in the flow hood. This is just an example of me transferring little seedlings from an intermediate flask to a final flask. It's my kid's bedtime. Let's break it down. I have gloves and a face shield, not absolutely necessary, but a good idea. If I'm singing at the top of my lungs, the face shield helps my breath not get everything gross. Only the sterilized tips and forceps, tips of my forceps are contacting the plants or the media. The vessels are staying away from the front edge of the flow hood. The front edge is where the dirty air is starting to mix up with the nice clean air. You keep your vessels away from that. Your hands, body, and face are all downstream from the vessels. So you see the flow of air coming from your filter. You want to work towards that, and you don't want anything dirty in between your open vessels and that nice clean air. You're popping off the lids. Now you want to set them with the clean side facing up. You do not want any of the inside of your containers, including the lids, to touch anything dirty, such as the surface of your flow hood, even though hopefully it has been wiped down with 70% alcohol. That just helps keep everything in there clean.
Caleb,
Yes?
why are you wiping down with 70% alcohol rather than 95% ethanol?
It's a good question. I think I know the answer.
Other than maybe cost.
Right, yeah, you can buy 90, dilute it down, you make it last longer. It's my understanding that 70% is more effective at killing microbes because it allows that alcohol to penetrate through like the bacterial cell membrane and actually destroy the inside of the bacteria. 90% sort of cauterizes the outside of the bacteria, and it still has a chance to like break free and grow again.
Because everybody I've seen was using 95% ethanol.
Yeah, you should use 70. I don't know.
I can maybe add something as a microbiologist.
Oh, awesome, yes.
We were always taught that 70% alcohol is optimal because it doesn't evaporate as fast. 95% alcohol evaporates too fast and
Okay.
not nearly as effective as 70%.
Thank you. Yeah.
The other thing I'll add to that is that they also say that it's not really completely understood why ethanol at these different concentrations completely kills bacteria or microorganisms. There's probably many different factors. But the other component is that the outer membranes of those can be a little more hydrophilic, and if you don't have enough water content in there, you can't lyse them open. But you do want a high concentration of ethanol because some of the mechanism, I think, is the osmotic pressure of the water inside the bacteria flowing out to the ethanol is what ruptures the membrane and breaks them open. So it's kind of that balance, and almost every lab you work in would recommend 70% over higher concentrations.
When I was in school to wipe down benches, we were still using carbolic acid.
Oh, wow.
And your fingers never touch the inside of the vessels. You send me seed pods, I touch the seed pod. I never touch anything ever again until it's in your hands. That's the only thing I've ever touched with my hands is the outside of that seed pod. From then on, I'm working with forceps, scalpels. I never touch a single plant that you receive from the lab. I know them, just not intimately. Green pod sowing. This is a pretty common form of sowing orchid seeds. The outside is disgusting. The inside is hopefully sterile. So you only need to sterilize the outside of the pod, cut it open, get those seeds onto your media. How do you clean the outside of the pod? Start by lighting it on fire. You can dunk it in alcohol and then light it on fire. It's not completely necessary, but it is fun. Then you soak it in, I like to use 20% household bleach plus a little bit of soap. Helps get it in contact with everything. Shake it up. Give it at least 10 minutes. At least 10 minutes. That should kill everything on the surface. And then you take it into your flow hood or your still air box. I like paper plates as a working surface. You can stack them up, throw them in a bag, the right kind of bag, or aluminum foil. You can cook them. They look disgusting. They're 100% clean. I reuse them. You're going to use sterile technique to cut this pod open on those sterile plates. And then you tip that pod upside down and you let the seeds fall onto your media, hopefully nice and evenly distributed. Sometimes those seeds don't want to leave that nice cozy womb, so you got to tap your forceps with your backside of your scalpel, and that helps get them to float down onto your media. Dry seed sowing. Some flasking services won't even touch dry seeds. I love dry seeds. Send me your dry seeds. Remove any little bits of pod or other debris from the seeds. Sometimes you even find little bugs in there. I usually let the grower know if I find some bugs. Hey, you seem to have a two-spotted mite problem. Then you add your seeds to a syringe. I like to use a 5 to 10 milliliter syringe. You put the cap, you get nice syringes with little caps on them, real cheap, Amazon again. Add your 10% bleach solution or your NADCC, which I love. You add that to the syringe plus a drop of your surfactant. You shake it up. Get your seeds nice and evenly dispersed back in that sterilizing solution. I like to pull the plunger back on that syringe. It kind of creates a vacuum. Gets that sterilizing solution right up there against the seeds. Sometimes it even gets through the cell walls a little bit, gets everything nice and clean. The total time in your solution should be about 10 minutes for your 10% bleach, or about 20 minutes if you're using the NADCC.
Do you do it longer for paps?
Tools of the trade. You continue agitating them until you're about a few minutes from the end, and then you allow those seeds to settle to the bottom of the solution. Viable seeds typically sink. PAFs might not. Now you're going to make sure that you're working in your flow hood or your still air box. You depress the plunger to expel that sterilizing solution while keeping the seeds inside the syringe. Wear gloves for this, guys. I don't know who this fool is. He's going to get bleach all over his fingers. Using a paper towel at least. Then you're going to draw in some nice sterile water or really diluted down NADCC solution back into the syringe. And then you'll probably want about a milliliter of solution per petri dish that you plan on sowing. So if you're going to sow 10 petri dishes, pull up 10 milliliters of your sterile water. Make sure you have enough seeds. Agitate that before you dispense your milliliter onto each petri dish. Tilt, swirl your petri dish to nice and evenly distribute them. Replace the lid, seal it up. Place them under nice, weak light for germination. Transitions. You're going to start with a mother flask. Probably have an intermediate flask and a final flask. When you move them from mother flask to intermediate flask, typically when you can work with them, they're going to be tiny. Once they're big enough that you can work with them, you move them up. Then you want to move them again from your intermediate to your final. Before the roots become overgrown and all tangled up, it'll become a big mess. It'll really slow you down. And then final flasking to deflasking is when the plants have filled out the flask really nicely. The roots are really well developed. The leaves haven't quite touched the top of your vessel. The total time from start to finish is usually between 9 and 18 months. Really depends on what you're growing. A lot of people don't do an intermediate flask. I like to. It just gives me another chance for quality control. Not all of these little protocorbs, not all of your seedlings are going to be great. They're not all going to be vigorous. They're not all going to be well formed. I'm being highly selective when I move from one step to the other. Only the best, most uniform, best growing, most well-developed, non-mutated orchid seedlings are ever going to make it into the final flask and then onto my customers. Assuming I have enough. Sometimes I get just a few seeds, then I'll contact my customer and I'll say, hey, we don't have a whole lot to work with here. Do you want me to just make as many as we can? They might not be at the best quality, but you will get plants. And then they let me know whether or not to bother with it. Yeah?
When you have an ugly duckling or a terrible one, do you keep them to yourself to see what they do, or you just pitch them?
No, these aren't my plants. Anything that doesn't go to the customer goes, it gets killed. People spend a lot of time breeding their orchids. They're developing just the right kind of plant. They've got big hopes for these plants. They're not mine, you know? So I don't keep anything. Yeah?
So that was actually a question I was gonna ask later. So if somebody spends 30 years of their life, finally coming to this final plant, and they send you the seed pod, do you have a professional contractual relationship with them? Do you have to carry, malpractice insurance or something that would, or E&O or something that would, I don't want to need to get really into the weeds, but, there's nothing to sue if it's just you and a forceps and a, what do you have that assures a client that you're not going to share their 30 years of life work with 50 other people, other than your own promise?
That's a great question. I should look into that. I have heard of people who flask who have gotten caught, like selling material on eBay and stuff. I have looked for all the other flasking people that I can find in the country. There's not very many. I've never seen one who really signs a hardcore contract. It might be more common for Mary cloning when you're doing lots of clones. Are you a lawyer? You want to help me out? Pro bono?
Financial analysis and that's my background. So pricing that insurance plan from a statistical point of view is what I did for a living. So
Cool.
I was wondering what kind of weird insurance you carry, because you're dealing with somebody's life work.
Yeah, that is an excellent question. I am not super familiar with legal stuff, and I feel like that would be a great way to set your business apart, to have a nice contract that people can feel safe and secure with, rather than my word. Maybe I should talk to you after this.
Well, that's why an architect carries, an E&O insurance, because there's nothing to sue. It's a desk and a computer and
Yeah,
frame.
right.
You have to have something, to assure the client that, if things go pear-shaped, that
Right.
there's something to do. So,
And then you probably want something from the other end, because you might go under contract to grow a whole bunch of plants, and then they just say, no thanks, I don't want them. You just spent 18 months, and now what, right?
I would comment that on the flip side, a flasking agency or someone you're working with, their career is, their work. A lot of people, they're hard to find people to take flasks. We shopped around for quite a while before we eventually found Caleb, and it's an industry that thrives on people having accountability by their own stuff. But you bring up a good point.
If I sell someone's stuff and you get caught, you're out of business. It's a really stupid thing to do. Just don't do it. Yeah?
Alternatively, you might not want to have a contract saying that you're not going to, because then if, say, someone else has made something that looks really similar, then the people who hired you might be like, you sold my things, even though you actually didn't.
Sure. Yeah, you could get accused, falsely accused, I guess. Yeah. I
Didn't think you were gonna get it illegal.
no. And honestly, like most of the time, I don't even necessarily know what the plant even looks like once it's gonna bloom. I'm just like, just let me know the genre at least, so that I can make the right media. Beyond that, I don't really care. It's not my plants. Yep.
So
And then
who are either your or other Flaskers' clients? Are they botanical gardens, mainly individuals that wanted to?
I once got corrected, called them customers, not clients. I don't know why. He had a really good reason, so I call them customers. My customers are hobbyists, professional growers. That's mostly it. I could reach out to like nonprofit organizations, see if they want to do that. I'm still working on building my customer base, but right now it's mostly hobbyists with smaller orders, and then sometimes like commercial growers, and they're gonna want like 30 flasks of each different thing, which is a lot of plants. 30 times 25, you know.
How much does it cost to hire someone like you to make like a hobbyist flask?
Go on my website. It takes about the same amount of time to make 1 or 2 flasks as it does to make 10 or 30. There's just a lot of initial work that goes into it, so it's a little more expensive when you're talking like 4. 4 is like, to make it even worth my time for me to make any kind of money at all. So 4 to 10, I charge like $30 to get the seeds established, and then $30 per flask. Some people have gotten in before that higher price, and they're grandfathered in. But then if you're asking for like 10 flasks or more, I drop the price down to $20 to initiate the seeds, and then $22 per finished flask. That equals out to about $1 per plant. It's not much. I think it's a pretty good deal. There are people who do it a little bit cheaper, but I pride myself on good customer service. I let you know when I got your seeds, when it's sown, did they germinate, when I put them up into the next flask, when they're about ready. I've heard, and maybe it comes with us introverts working in the lab, we're not always the best at communication, so I've heard of people sending seeds off to the lab, they never hear back, and then they have to call them. What's going on? How are things going? Oh, sorry, your seeds all got contaminated, stuff like that. That communication does take time, so I charge a little bit more because it takes time, but I like to think that the customer appreciates knowing what's going on.
I'll give another comment on that too. That I've had seed pods that we've sent off to Caleb, and he warned me ahead of time, right, that these don't look great. The seeds are not probably fertile, but you want to go ahead and seed them anyway. Again, knowing that he's going to charge us the full amount for them, but he does do that ahead of time of actually looking at the pods and making sure the seeds are viable before just trying to start everything else and just taking the client on it. So I appreciate that.
I appreciate you. So deflasking, that's at the end. It's best done when the plants have grown into their vessel. Their leaves are nice and large, but they haven't quite touched the top. That can kind of stunt their growth. And then they have nice, big, extensive roots at the bottom. Deflasked orchids are very sensitive due to the unique growing environment that is in the flask. They have poorly developed leaf cuticles. The cuticle is like the waxy coating on a leaf that helps keep it from desiccating, from drying out. They don't develop a very good one in the flask. Their stomata, which are like their little pores on their leaves that help them breathe and exchange gases, those are often locked open from being in the flask at 100% humidity all the time. So that, again, leaves them sensitive to drying out. And then the leaves are adapted to low light levels. In the lab, it's often like 1/10 of sunlight. So they're sensitive to sunburn. And so people ask me, how do you deflask these orchids? Well, for one, I don't have a greenhouse. I grow them in a flask. Not that interested past the flask, but I have done some experimenting just so that I could provide a better customer experience. The problem is due to the different conditions that you all grow your orchids in, there's no like one size fits all for when I hand you the flasks and you take them out. You might be going in a greenhouse with high humidity. Maybe you're growing indoors with low humidity, but hardly any air circulation. Generally, you need to provide a well-drained but moisture-retentive growing mix, reduced light levels compared to your mature plants, above average humidity, but you still want to maintain some air circulation so that you don't get mold. I'm popping out some orchids. This is a customer who actually likes me to send them to him deflasked. He's in Hawaii, so shipping would be outrageous if I sent them to him in a flask. This allows for more compact shipping. I gently remove the orchids from the flask. I like to use a nice big open-topped flask so you don't have to break something. You don't have to try to pull a ship out of a bottle. You gently remove them from the flask. You rinse off all that agar media. It's highly nutritious. Remember, you don't want to grow microbes. They'll still grow outside of the flask especially. Then you inspect your plants. You remove any media that's still clinging to the roots as best you can without damaging the roots. And then you want to remove any dead material that could encourage mold. I like to use a nice little leftover strawberry container. Clean it really well. Here I'm using a potting mix made up of Orchiata classic size. It's kind of smaller. And then I've chopped up some sphagnum moss for a little more moisture retention. I'm growing these indoors with pretty low humidity, so that's why I'm using something with a lid. I'm using media that retains moisture a little better. I put down a layer, and then I put my plants on top, nice and evenly distributed. And then I cover up the roots with the media. I make sure not to cover the crowns. That can encourage mold. Where are these? These are Solestis. Yeah, it used to be something else. Now it's Vanda Solestis. Rhincostylus. Yep. Some seeds I purchased myself just for this. You water them in nicely. You close the lid if you're using a lid. Go ahead and use your regular orchid fertilizer. These things have been growing in nutrient-rich media. They can handle some fertilizer right off the bat. Put them in your grow area and then inspect them frequently. They're fragile. Over the course of a few weeks, you slowly expose them to a little more light, a little less humidity. Once they've established themselves well, maybe they've got a couple new leaves growing or some nice big long roots, then you can pot them up individually. All right, do we have time? Forgot to start my timer. Okay. So harvesting seeds, let's say you don't want to do this all on your own and you'd rather just send pods to the lab. The pod on the right is nice and plump. It's begun to change color. It's a good indication that it's ready. The pod on the left could have been allowed to mature a little bit longer. The AOS website has a really good guide for pod harvest times for the most common genre and hybrids. I still recommend giving them a bit more time than what this list says, if possible. If you're shipping your green pods, it's best to wrap them up in something like coffee filters or a paper towel. It needs to have some air circulation, but you also want to contain those seeds just in case your pod splits in the mail. Wrap it up snugly, you tape it shut, make sure you label it. Most flasking people are going to give you a code, or you're going to give them a code. Most of the time I don't even want to know what your orchid is. Just give me a code and the genre, I'll do the rest. If you want to harvest dry seeds, it ensures that the seeds have reached full maturity. If you send me a pod that's too young, I can't do anything with underdeveloped seeds. You send me dry seeds, I could still work with those. So it avoids the guessing of timing for green pods. On the other hand, your seeds could be lost if it splits open when you're not looking. So, pick your poison, I guess. It's best to harvest the pod as late as possible, but before it splits. This one split open in the mail. Luckily, they had wrapped it up really nice and tight. I unwrapped it. Oh, look, there's the seeds. I can work with these. Dry seeds, an advantage is they can be stored for an extended period of time. I'm going to show you how to do that. So if you're not quite ready to send them off to the flasker, you can just store them until you're ready. And then shipping dry seeds is also less of an issue in freezing weather. If you're sending a green pod in freezing weather and it freezes, it's just mush when I get it, and that's almost impossible to work with. You can put them in a little microcentrifuge tube, your dry seeds, or you wrap them up, do a little origami, or wrap them up in a paper towel. Securely contain them, and then you can store them over silica gel or calcium chloride, which is like damp rid, available at Walmart. Just an airtight container, throw them in the fridge. As long as they're dry and mature, most orchid seeds can actually be stored for several years this way. Please don't put your dry seeds in a plastic bag. Static, they cling to the inside of the plastic bag, so hard to get out of there. I've had people send me seeds in an envelope, and good idea, but they can get out. So then, logically they think, well, I'll tape any little place where they can get out. Problem is, now I get the seeds, all the seeds are stuck to the Scotch tape. Thank you. And if you're in need of any flasking services, Flask Factory, oh my gosh, the formatting, theflaskfactory.com, and then my email is caleb@theflaskfactory.com. I do have business cards up here if you want one. And I've got a nice little finished flask here. This is just like ready to ship out, just about perfect. Those are nice little Paphiopedilums. Looks like some kind of Maudier. It's got nice mottled leaves. It's probably about 9 months right there. These are some protocorms. They just finished germinating. Just tiny little babies. And then a couple examples of orchid seeds. If you've never seen orchid seeds, they're like dust. 2 different kinds here. Some of them come with like cool colors. I've seen pink, green, usually brown and beige. But thanks again. Yeah.
Do you have any trouble, like you mentioned, you have one client in Hawaii. Do you have any trouble shipping to Hawaii or certain states like Arizona that have certain restrictions?
Yeah, Hawaii is great because they have a really big industry of growing orchids, and I think they try not to get in the way too much. I label the outside of the box deflasked orchid seedlings, and they pretty much let it through. Some states like California, for instance, might require them to be in the flask so they can ensure that they're sterile. There might be some paperwork involved. I've actually had the luck of not having a California customer yet. So, but yeah, it's good to look into each state and see what their regulations are around plant material. It's a lot more challenging if you're talking international. I don't even deal with it yet. Maybe someday I will. Then you need like phytosanitary permits. You might have to deal with CITES because some of these plants are, rare. They may be on some list. So, yeah, but. Mm-hmm.
Have you ever purposely added a symbiotic fungus to your other method of your flasking from a sterile environment just to see if that would do anything?
Have I ever intentionally added it to what?
So when you've done a dry flasking or dry seed, or just added the fungus both at the same time?
Usually if I'm doing symbiotic, I don't do it much anymore. It's usually for conservation efforts. A lot of times I was working with dry seeds. I'm not quite sure I understand the question. But yeah, you take the seeds. They could be dry or they could be from the pod. You put them on that oatmeal-based media, and then you actually do add the fungus with those seeds, and the fungus grows out, infects them, and then they grow. Sorry, did I answer your question? Okay, I'm sorry. Any other?
Thank you very much. Yeah, thank you. I appreciate it.